Welcome back to this next video in which we are discussing influenza. Okay, so we've just been discussing the adaptive immune response that is initiated in response to influenza viral infection. So, to begin this video, I would just like to begin by briefly summarising what we have learned about the adaptive immune response one last time, because it's a complicated topic, immunology, so I think it's worth repeating everything that we've seen one last time. So, we have seen that in response to influenza, you're going to initiate two arms of the adaptive immune response, the two major arms of the adaptive immune response. You're going to initiate a B-cell response, and you're also going to initiate a cytotoxic T lymphocyte response. And remember, the B-cell response is to destroy and attack extracellular influenza viruses, and the cytotoxic T lymphocyte response is to attack the intracellular influenza viruses. So let's start off with the cytotoxic T lymphocyte response because that one's simpler. So overall, in this response, you produce loads of cytotoxic T lymphocytes or effector CD8 positive T cells that are going to be directed against fragments of the antigens of influenza viruses. Now, these are going to kill virally infected cells, and the way they're going to do this is that all nucleated cells continuously sample proteins from their cytoplasm and break them up into little peptide fragments and present them on loads of MHC class I molecules that they have on their surface. Now, when a cell becomes virally infected with influenza, then it will do this for the influenza antigens as well as its normal proteins, and therefore it will end up with some MHC class I peptide fragment complexes containing peptide fragments of influenza viral antigens. Now, if a cytotoxic T lymphocyte finds a virally infected cell with an MHC class I peptide fragment complex, where the peptide fragment is the one that it's directed against, it will bind to that cell using its T cell receptor, and then it will kill the cell using perforin granzyme mechanism and the fast ligand mechanism as well. And that is the cytotoxic T lymphocyte response, and that's going to be happening at the respiratory tract epithelium, and respiratory epithelial cells that are infected with influenza virus are going to be destroyed. They're going to commit apoptosis, and they will destroy all of the influenza virus stuff that is inside of them. Excellent. So that is to destroy intracellular influenza viruses. Now let's talk about how we're going to get the extracellular influenza viruses. And as we'll see, the B-cell response, it's not just extracellular. I mean, we've talked about antibody-dependent cell-mediated cytotoxicity, which is going to help with intracellular clearance as well. But majorly, this is the extracellular response. Okay, so what you're going to do in this response, remember this one's more complicated, is firstly you have to produce a population of effector CD4 positive T cells that are also called helper T cells, and the specific type that you're going to produce most of in influenza viral infection is the follicular helper T cells, which are very diligent at helping with B cell activation. The other types that you can produce are important in other types of adaptive immune response that aren't relevant to us, so cross these ones out. So you're going to produce a whole host of follicular helper T cells that are going to be again directed against fragments of the antigens of the influenza virus. So we produce loads of, and we haven't really got a picture for them. I will just put in a little picture here of our follicular helper T cells. So here we go, in purple here. This can represent a follicular helper T cell. So we're going to produce a whole bunch of follicular helper T cells which are directed against peptide fragments of the antigens of the influenza virus. Okay, and these are going to help with B-cell activation. So now let's talk about B-cell activation. And so overall, as part of the B-cell activation process, you're going to activate B cells that are directed against epitopes of the antigens of the influenza virus. Now, the way, remember, that this occurs is that the B cells have to receive help from follicular helper T cells that are directed against peptide fragments of the same antigen that the B cell is directed against an epitope of. So this is the purpose of the follicular helper T cells, and also the purpose of other helper T cells if we were to create those for B cell activation. So in order to get B cell activation, I'll say this one more time, you have to have the B cell receiving help from helper T cells that are directed against a peptide fragment of the same antigen that the B cell is directed against an epitope of. So overall, with the follicular helper T cells, we're now going to get activation of B cells, and I realise that when this is copied, it's lost an arrow there. There should be an arrow here. So overall, we're going to get activation of B cells that are directed against epitopes of the influenza viral antigens. And of course, we'll get lots of different B cells being activated, directed against different epitopes of potentially different influenza viral antigens. Okay, then these B cells will differentiate into the effector B cells, the plasma cells. So overall, with the B cell response, we're now going to get these plasma cells, which are going to be directed against epitopes of the influenza viral antigens. Now, when each of the B cells decides to differentiate into a plasma cell, it has to choose a type of antibody that it's going to produce. There are different types of antibody that it can produce. It, of course, has its set antigen binding region, but the constant region of the antibody molecule, it can choose a design that it wants to use, and the different antibody designs are useful against different types of pathogen. Now, in the influenza virus immune response, we're going to mainly produce IgG and IgA. We'll produce a little bit of IgM. We will not be producing IgE. So, overall, we're going to get loads of plasma cells that are directed against epitopes of the antigens of the influenza virus, and some of them will be producing IgG, and some of them will be producing IgA, and a tiny number will be producing IgM. Now, the antibody molecules that they secrete once they're in the bone marrow, what do these actually do? Well, IgA is going to be secreted onto epithelial surfaces, so important for us, it's going to be secreted onto the respiratory epithelial surface. IgG is going to go into extracellular fluid all over the body, but particularly it's going to go into the exudate, so it's going to arrive at the respiratory epithelium as well. IgM will remain within the blood. Now, one of the major ways that antibodies help to fight the extracellular viral particles is they neutralize the virus particles. So when we have antibodies directed against epitopes of the hemagglutinin or hemagglutinin esterase fusion proteins, these will bind onto them and they will block the binding site of that protein for the sialic acid residues, and therefore the virus particles coated in these antibody molecules are not going to be able to actually infect cells anymore, so they're not dangerous, and that's called neutralization of viruses. So that's a major way that we can fight the extracellular virus particles. So antibodies are incredibly powerful at doing that. In addition, we're going to get opsonization of the virus particles by, this is IgG that's going to do this rather than IgA and IgM. So what will happen is we will produce IgG molecules against epitopes of the surface antigens of the influenza virus particles, and this means the influenza virus particles will end up coated in IgG molecules. And macrophages, neutrophils, and dendritic cells, the phagocytes, have a receptor, the Fc gamma R3 receptor, for the constant region of IgG molecules, and when they find a particle in the extracellular fluid that's coated in IgG molecules, they say this is obviously a pathogen, let's phagocytose it. So there's another way that the antibody molecules are going to help fight pathogens that are extracellularly. Not only have they neutralized the virus particles, but they're now leading to the phagocytosis of the influenza virus particles. Finally, the antibody molecules are also going to actually help fight intracellular influenza viral particles as well. So it's not just the extracellular influenza viral particles that are going to be attacked by the B cell response. We know, of course, that infected cells are covered in the influenza viral surface proteins, so the antibodies will bind onto those surface proteins on the surface of the cell, and again, the IgG ones are the significant ones. Natural killer cells have a receptor, this Fc gamma R3 receptor, again for the constant region of the IgG molecules, and therefore when they find virally infected cells that are coated in IgG molecules, they will then kill those cells through the perforin granzyme mechanism. So, the B cell response, obviously I stressed at the beginning that mainly it's going to help us deal with the extracellular virus particles, whereas we've got the cytotoxic T lymphocyte response for dealing with the intracellular influenza viral particles, but actually it also helps destroy the intracellular influenza viral particles as well. Excellent. So this is the way that the adaptive immune response is going to annihilate influenza from the respiratory tract epithelium. Here is how we get rid of the extracellular influenza viral particles by helping the cells of the innate immune system destroy them. We first neutralize them and then help the immune cells of the innate immune system find them and destroy them. And we also get rid of the intracellular ones through this mechanism here and also through the cytotoxic T lymphocyte response. So, there is the adaptive immune response to influenza. I hope you enjoyed that. Now, let's talk about what's going to happen after we have cleared the influenza viral infection from the respiratory tract epithelium. How is the thing going to turn off? The adaptive immune response does turn off afterwards. And then I want to talk about how do you get long-lived immunity to influenza virus strains. So, let's firstly go on about how the thing turns off. So, these effector cells that we have seen, so the effector cytotoxic T lymphocytes, the effector CD4 positive T lymphocytes, and the effector B cells, the plasma cells, they're all reasonably short-lived, particularly the cytotoxic T lymphocytes and the effector CD4 positive T cells, so the helper T cells. And so after a few weeks, these cells will start to die, so the effector cells will start to die. The plasma cells will also start to die, however some plasma cells will live for longer. So I'm just going to sort of write a little bit of this down. So, where should we go for this? We'll go up to here. Okay, so within a few weeks, so I'll put few weeks, what's going to happen is these effector cells of all the different types that we've seen, so the effector B cells, the effector CD4 positive T cells, and the effector CD8 positive T cells, so within a few weeks, the effector cells die. So they're no longer going to be useful, and that's fine because they've done their role, they've cleared the infection by that time. So the effector cells die. Now, this is what I've been talking about all along, the motivation for why we need the memory cells, why we needed the memory CD4 positive T cells, the memory CD8 positive T cells, and the memory B cells. Because if we didn't have those, the effector cells are just going to die and then we'd lose the entire clone that was actually useful to us. The memory cells live on. They will live for years and years and years, if not your entire lifetime. And so, but in a few weeks, the effector cells will die. Now, the exception to that is some of the plasma cells live longer. So you do have some long-lived plasma cells. So I'll just put here, but you have some long-lived plasma cells. However, they do not live that long. So they might last for a few months rather than a few weeks. So long-lived plasma cells. So I'll put here few months, and these will remain in the bone marrow producing antibodies which they'll be putting into the bloodstream. Now, why is this significant then? Well, this is significant for when we want to talk about how you get long-lived immunity to influenza virus. So let's say we have been infected by a certain strain of influenza virus of one of the three species. So we've got some strain of influenza virus that has infected us. We have launched a very powerful adaptive immune response to it. It has cleared the influenza virus from the respiratory tract. And now what will happen is all the effector cells will start to die, but for a few months afterwards you're still going to have these long-lived plasma cells in the bone marrow producing antibodies against the proteins, the epitopes of the antigens of that influenza viral strain. And the two major types that we've discussed you'll produce are IgG and IgA. Now IgA is very significant here because that's going to be put onto the respiratory epithelium. So, after you've cleared the influenza viral infection from the respiratory tract, what will happen is the respiratory epithelium will then heal, the basal cells will have their work cut out and they'll repopulate the ciliated columnar epithelial cell population. Then, after that, on the surface of the respiratory epithelium, once it's refunctioning again, you'll get lots of IgA that's still being produced by these long-lived plasma cells in the bone marrow put onto the surface. So, IgA is going to be important, and this initially mediates your lasting immunity to that influenza viral strain. So for the first few months after having the influenza infection, you're going to have immunity that is really, really good against that influenza viral strain, and also other influenza viral strains that have the same hemagglutinin serotype. So this is important. This, of course, varies depending on whether we're talking about influenza A or influenza B or influenza C. So, let me explain what I'm trying to get across here. For a few months after you have had the influenza infection, you are going to have, in the mucus on the surface of the respiratory epithelium that has now healed, IgA molecules, and some of these IgA molecules will be directed against the hemagglutinin proteins of that viral strain, or the hemagglutinin esterase fusion protein if we're talking about influenza C virus. So I'll just write this down here. So, some of these IgA molecules in the mucus now, on the surface of the respiratory epithelium, will be directed against hemagglutinin if we're talking about an influenza A virus or an influenza B virus strain, or they'll be against hemagglutinin esterase fusion if the infection was an influenza C virus strain. Okay, and this protects you really, really well. So for a few months you have an absolutely fantastic protection, and this is the mechanism that gives you lasting immunity for a few months anyway. The mechanism is then going to change for what gives you lasting immunity after those few months. So we are now discussing oh whoops, I've misspelt that. How can I add that in? Imagine it says lasting immunity. Okay, so for a few months after you've had the influenza infection, you're going to have IgA molecules in the mucus on the surface of the respiratory epithelium that are directed against hemagglutinin. Obviously, you'll have IgA molecules against other influenza antigens as well, but the important ones are that it will be directed against the entry protein. Hemagglutinin if we were talking about influenza A or influenza B, or hemagglutinin esterase fusion if we were talking about influenza C virus. Now that means that if you were to then attempt to get infected again, so if you were to breathe in this same strain of influenza virus again, you would not get the disease again. Why? Because the influenza virus, of course, would end up in the mucus, and then in the mucus there's going to be loads of IgA, which are going to bind to the entry protein, whichever one of these it is, and neutralize the virus particles. So I hope I've communicated what I wanted to communicate here, that for a few months after the infection, your lasting immunity to the influenza virus will come from the fact that in the mucus you have IgA molecules directed against its entry protein, whether that's hemagglutinin or hemagglutinin esterase fusion, which will neutralize it as soon as it lands on the epithelium, and therefore will make it impossible for that virus particle to then infect the ciliated columnar epithelial cells. So this is the way that you get lasting immunity for at least the first few months after having the influenza infection. Now, this will go beyond just the strain that infected you. Remember, influenza A virus, influenza B virus, and influenza C virus, all of them have a huge number of different strains. You'll have been infected by a specific strain. However, remember, as long as the new influenza virus that's coming in is of the same hemagglutinin or hemagglutinin esterase fusion serotype as the original viral strain that infected you, the antibodies will have a significant cross-reaction with this new strain's hemagglutinin or hemagglutinin esterase fusion proteins. So it will actually protect you against more than just the strain that infected you. So, if we're talking about influenza A virus, we know that there are 16 different serotypes of hemagglutinin molecule. So, what will happen is for a few months you will be protected against all of the strains of influenza A virus that have the same hemagglutinin serotype as the one, the influenza virus strain that infected you originally. For influenza B virus, we know that there is only one hemagglutinin serotype there. So what will happen is you will get immunity against all, or at least most of them. It will, of course, depend on just how similar the hemagglutinin proteins are, but as long as they're similar enough between the two different strains, the strain that infected you originally and the strain that's now coming to infect you again, is as long as they're similar enough, then there will be cross-reaction between the antibodies, and therefore the IgA molecules that you produced against the original influenza virus strains, hemagglutinin molecules, will be able to bind to this new strain's hemagglutinin molecules. At least some of them will be able to, and therefore you'll be able to neutralize the new strain that's infecting you. And the same for influenza C virus. Remember, there's only one serotype of hemagglutinin esterase fusion. Okay, of course, for influenza A virus, there are different serotypes of hemagglutinin, and hemagglutinin molecules of the different serotypes are very, very different. So, let's say originally you got infected by H1N1 serotype of influenza A virus, then if the next time you breathe in influenza viruses, it's H2N2, let's say, then the IgA molecules that you have in the mucus from the H1N1 influenza A virus strain infection will not be able to protect you from this new influenza H2N2 strain, because the hemagglutinin molecules are too different, and therefore you're not going to get neutralization in the same way. So because of the huge variety of influenza A virus serotypes for hemagglutinin, you won't be protected against all influenza A viruses, whereas for influenza B and influenza C, you'll be protected against a lot of them for these first few months. So this is how lasting immunity works initially. You have this IgA, and some of the IgA molecules will be directed against the entry receptor of the influenza virus strain that infected you. Now, that means that if you breathe in that same influenza strain again, you will absolutely be protected against that, because loads of these molecules will bind to the entry receptors of that influenza particle that's come in and will neutralize it so that it cannot infect cells. But the protection will go beyond just the mere strain that infected you. It will also cover other strains of influenza virus, of all three of the species, that have similar entry receptors. So, as I've said, if the original infection was some strain of influenza A virus, then the other strains of influenza A virus that are of the same serotype you will be protected against. For influenza B virus, there is only one serotype of hemagglutinin, therefore, for a huge number of those influenza B virus strains, you will be protected against them, because the IgA molecules will be able to cross-react. They'll be able to react with at least some of them will be able to react with epitopes that are shared between the different strains of influenza B virus, and the same for influenza C virus with the hemagglutinin esterase fusion. So this is where lasting immunity from influenza comes from for the first few months. Unfortunately, it does not last. The long-lived plasma cells, they will start to decrease in number. They do start to die. And as the number decreases in the bone marrow, the amount of IgA they're producing as a population will go down and down and down. So therefore, the amount that's in the mucus of the respiratory epithelium will go down and down and down. And as it goes down, that will mean that it's less likely that if you breathe in an influenza virus particle, it will be completely neutralized by those antibodies. You need to have a certain high concentration of IgA in the mucus against the hemagglutinin in order for it to be neutralized. Okay, so after a few months, this will wear off. So there's a new mechanism then for lasting immunity, and this is the secondary immune response, which I want to just briefly discuss. So, what we have just discussed is what we would call the primary immune response. When you initiate these B-cell responses and T-cell responses for the first time, that is what we call a primary immune response. Okay, those clones of B-cells and T-cells directed against, if it's a T-cell, a specific peptide fragment, and if it's a B-cell, a specific epitope of an antigen, when they've been used for the first time, that's the primary immune response. When we use those clones again, i.e. those specific designs of B cells and T cells again, that is what we call a secondary immune response. So, again, let's do this by talking about an example. So let's say we have an example. Let's say we were infected by a strain of influenza A virus that had the serotype H1N1, such as, for instance, swine flu. The pandemic of 2009 was serotype H1N1. So we've been infected by a strain of influenza A virus of the serotype H1N1. We launch a primary adaptive immune response against this. It annihilates it, and then for a few months we will have IgA molecules against the hemagglutinin of the serotype H1 in our mucus of our respiratory epithelium. However, if we get infected years later again with this same virus, let's say, what will happen now? Well, it's not going to cause as bad an infection as it did for the first time. However, it is still going to cause an infection because we can't neutralize the virus particles as soon as it arrives anymore because we don't have enough IgA anymore on the surface of the mucus. So what will happen is the infection will proceed as we've described in this video. It will infect the ciliated columnar epithelial cells beginning in the nasal epithelium. An inflammatory response will be initiated, and then we'll want to start the adaptive immune response. Now, because we have used the clones of B cells and T cells previously, it will be a secondary adaptive immune response, and this will be much quicker and much more powerful. And the reason that it's going to be much quicker and much bigger is because the cells that we will be looking for, the CD4 positive T cells, the CD8 positive T cells, and the B cells that we will be looking for, the antigen presenting cells and also the actual antigen proteins themselves in the case of the B cells, to activate, there's going to be far more of them. And this is because of the memory cells. So we might have started off in the primary immune response with just having one B cell of this clone. We've replaced it with loads of them that are performing its role. So it's going to be far easier to actually activate this B cell response because it's going to be far easier for the influenza viral antigen to just happen to come across one of the right B cells with a design of immunoglobulin that can bind to its epitope. So the amount of time it took to activate this B cell response because you were waiting for one of the correct influenza antigen just to happen to bump into this B cell, that will be much shorter in the secondary immune response because we have far more of these cells. And it's the same with regards to the T cell activation, where the antigen presenting cells are looking for the T cells directed against the correct peptide fragment. We've got far more of those T cells now, so the time it will take for an antigen presenting cell to find one of those T cells will be much shorter, and therefore the response will be much, much quicker. So the secondary immune response is much, much quicker. Now, what does that mean? That means that the virus is not going to get as far along. Remember, the virus usually makes its way from the nasal cavity down to the nasopharynx, down to the bottom portion of the larynx, down into the trachea, down into the early bronchi, and then the primary adaptive immune response will kick in, destroy it, and it doesn't then get any further. But now we've got a secondary immune response which is going to come in much quicker. So it might come in when the virus has only just got maybe to the nasopharynx. So maybe you'll only get nasal symptoms, and then the secondary adaptive immune response will kick in, it'll bust the virus out, and it will never get down to the larynx. It'll never get down to the trachea. It'll never get down to the early bronchi. And therefore it will cause a much less severe illness because it's infected far less of the respiratory tract before the immune response kicked in and busted it out, is the point. Okay, so going over the symptoms that you might get in this second time of being infected, here are the symptoms. You'll get the nasal symptoms, so you might get a runny nose, nasal congestion, sneezing. You might get the sore throat because it's got to the nasopharynx, but you won't get the hoarse voice, you won't maybe get the cough, you won't maybe get the shortness of breath. And because you've got much less respiratory epithelium infected, and therefore much less respiratory epithelium inflamed, you're going to have much less TNF-alpha, interleukin-1 and interleukin-6 going into the bloodstream, and therefore the systemic symptoms will be much less bad. The more respiratory epithelium is infected, the more is inflamed, the more these molecules you get, the more the systemic symptoms you get. Okay, so my point is that this illness is going to be much less bad when you get it for the second time because the adaptive immune response will come in so much quicker. And indeed, you might class this now as a cold clinically. So although biologically we like to think of flu as being caused by an influenza virus and colds as being caused by rhinoviruses and coronaviruses, clinically, as I've said, the difference between a flu and a cold is both of them have the local symptoms, the respiratory symptoms, but flu has horrendous systemic symptoms and makes you feel really, really unwell to the point that you can't function normally, whereas a cold doesn't do this. It doesn't make you feel really, really unwell. It doesn't give you anywhere as near as bad systemic symptoms. So even though you've actually been infected by a flu virus, because you've got the secondary adaptive immune response and you've seen this before, you might just get the symptoms of a cold, and therefore clinically we would class this as a cold even though it's caused by the influenza virus. So understand the difference between the clinical definitions of flu and cold compared to the biological definitions. Clinically, everything's defined in terms of symptoms, and if you've got the symptoms of a cold, even if it's been caused by the influenza virus, we call it a cold. Whereas, obviously, biologically, we like to think of flu as being caused by the influenza virus, but in fact, the clinical symptoms of flu only arise when you've been when it's this primary exposure to the influenza virus. So that is how lasting immunity will be given to influenza viruses, and how you will where am I going? Where is it? How you will have protection from getting full-on flu disease in the future when you get infected again by potentially the same strain of influenza that you had. Okay, in addition, what I want to add is that this will cover you against more strains than just the strain that originally infected you. So I was giving as an example, we had this H1N1 strain of influenza A virus. There will be a bunch of other strains of influenza A virus in that same serotype H1N1 that are very, very similar to that strain that we were infected with, and a lot of their antigens, so I might even just draw this, just so that I've got something to point out. So let's say we've got one strain of influenza A virus here, and we've got another strain of influenza A virus here. Let's say they both have the same serotype, so they are quite similar strains of influenza A virus, but they are different. So they're both H1N1, just to have an example. Now, if they're reasonably similar strains, then a lot of their antigens will be very similar and will have similar peptide fragments, indeed a lot of overlapping peptide fragments. So some of the peptide fragments of the antigens of this one will be the same as the peptide fragments of the antigens of this one. Of course, not all of them will, but there will be a significant overlap in their peptide fragments, and that means that the secondary T-cell responses, a lot of them can be used against this other strain that we used on this one. So when we initiated the primary adaptive immune response, when we got this one first, we will have created lots of memory T cells and memory B cells, and maybe a lot of those will be useful against this second strain that is similar, because a lot of its peptide fragments of its antigens are the same as this one. There's overlap in the peptide fragments of the antigens, so we can use those memory CD4 positive and CD8 positive T cells against this strain if we got infected by it, and some of the epitopes of the antigens might overlap between the antigens of this one and this one if they're very similar, and therefore we can use the memory B cells from the primary infection with this one against this one. So it's not just that you have immunity to the specific strain that you were infected with, you'll have immunity also, or at least a better immune response to other strains that are very similar to it. And that doesn't just go for influenza A virus, of course, that goes for influenza B virus and influenza C virus as well. Overall, once you've had flu once, you will have a much better immune response against that flu strain if you get it again than you had initially, and you'll also have a better immune response against strains that are very similar to that one as well, because of the overlap in the epitopes of their antigens and peptide fragments of their antigens, which means that you can use your memory B cells, your memory T cells that you got from the first strain against the second strain. Okay, so I hope what I have said makes sense. Initially, the lasting immunity is achieved through these IgA molecules, and that's fantastic. This is brilliant. This completely stops you getting the infection. Then afterwards, it won't completely stop you getting the infection, but it will deal with the infection much quicker, so it doesn't get as far down the respiratory tract, and you won't get anywhere near as severe symptoms. You might just get the symptoms of a cold, even though it is caused by influenza virus, and therefore will be clinically classified as a cold, not as flu. Okay, so I think we'll have a break here. In the next video, we will discuss antigenic drift and antigenic shift.